Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. So this is going to be my second video of the biochemistry series where I'm going to discuss more properties or more concepts pertaining to amino acids. So if you've seen my previous video, you would have seen about the classification of amino acids based on their R group. So the R group is nothing but your side chain. So if I draw it here, you have the amino group, you have the carboxyl acid group, and you have the R group here, which is going to be characteristic for each and every amino acid. You are going to have different names for the amino acids based on this R group. So there is going to be classification of amino acids based on their R group, and we have seen more about this in the previous video. Please do check it. So if you see that it is based on the polarity, that is the tendency to react with water at the biological pH. So the biological pH is close to 7, okay, close to 6 to 7. The reason why we choose biological pH, because that's going to be the real time scenario, like when you have to study about proteins or other biomolecules at a given biological pH, that is the pH that corresponds to the biological systems in an organism. So, if you see just a quick recap, the amino acids that fall into the category of non-polar aliphatic R groups are glycine, alanine, proline, valine, leucine, isoleucine, and methionine. All of these have R groups that are non-polar and they are aliphatic. Okay, so they don't exhibit any kind of polarity. If you see about the aromatic R groups, then we have three main important amino acids. They are phenylalanine, tyrosine, and tryptophan. And these aromatic amino acids are kind of exploited when we have to find the concentration of proteins. I'm going to discuss about it in more detail in the coming slides. Polar but uncharged R groups. So you have the R groups that are polar but they are uncharged, mainly because of certain electronegative atoms that are present in the R groups. So the amino acids that fall into this category are serine, threonine, cysteine, asparagine, and glutamine. So asparagine and glutamine, they have an amide group, and cysteine is a sulfur-containing amino acid, and serine and threonine, both of them have hydroxyl groups. Next, positively charged R groups. So these R groups contain a positive charge. So the amino acids that fall into this category are lysine, arginine, and histidine. Okay, and the negatively charged R groups mainly comprise of amino acids, aspartate and glutamate, also known as aspartic acid and glutamic acid. So the difference between asparagine and aspartate and glutamine and glutamate is the presence of carboxylic acid group instead of an amide group there. Okay, moving on. So the non-polar aliphatic R groups containing amino acids, so they tend to cluster together within the proteins and stabilize the protein structure through hydrophobic interactions. So we know that they are pretty much non-polar. So they tend to form the hydrophobic core within the proteins and hence they tend to cluster together within the protein. We all know that like likes like, which means that non-polar tend to cluster together and polar tend to remain together, non-polar tend to remain together. So in that particular concept, if you apply this, we know that if you suspend a protein in a polar solvent, all the hydrophobic amino acids or the non-polar aliphatic R groups containing amino acids will tend to cluster within the protein because they want to be away from the polar environment. Okay, and if you take glycine as the most prominent example, it has the simplest structure. We know the R group for glycine is just the H, so and it is also achiral. I would have explained this concept in my previous video. So it has two hydrogen atoms, so it's achiral. So it has the simplest structure. Its R group being very small, that is, the R group is nothing but your hydrogen, being very small, makes no real contribution to the hydrophobic interaction. It's just too small to make any significant hydrophobic interactions. So that is the reason why glycine is said to have the simplest structure. If you talk about methionine, again, you know that it's a sulfur-containing amino acid, and the R group is going to be non-polar aliphatic, and it has slightly non-polar thioether R group. Okay, the If you see the structure of methionine, it has the sulfur containing, and that thioether group, okay, is going to be non-polar. So if you see proline, proline is actually an amino acid. I would have explained about its structure in my previous video. So the aliphatic side chain with a unique cyclic structure, because it is an amino acid. Why? Because it contains a secondary amino group. Okay, so it contains a secondary amino group, and the secondary amino group is actually held in a very rigid conformation. It is not easily, it has a lot of steric hindrance, and hence there is going to be a reduction in the flexibility of the polypeptide regions that contain proline. So if you have a polypeptide and it has proline in some of its residues, then those areas of that polypeptide is going to exhibit a lot of rigidity, so it is not subjected to flexibility. Okay, so moving on to the next slide. Okay, so now I'm going to talk about the aromatic amino acids, that is the phenylalanine, tyrosine, and tryptophan. So all of them are relatively non-polar because they are hydrophobic as well, but all can participate in hydrophobic interactions. Even though tyrosine and tryptophan do have some polarity, there are groups. If you see the structure of tyrosine, so I'm just drawing it here. Um, so it has a H above, and this is the structure of tyrosine. It has an OH here. So it has a hydroxyl group in its side chain, and this can actually participate in hydrogen bonding as well. So this oxygen atom that is present as a part of the hydroxyl group in the R group can actually participate in hydrogen bonding. Why I am stressing on this particular property is that it plays a very crucial role in various enzymatic reactions. So I will be discussing about certain prominent enzymatic mechanisms in my upcoming lectures as well. So if you see tryptophan, it also has a nitrogen which is pretty polar in the indole ring of the tryptophan. So nitrogen is a nitrogen atom in the, so the spelling is wrong here, so it's nitrogen. Okay, the nitrogen atom in the indole ring of tryptophan is kind of polar, and hence makes tryptophan a little bit more polar. So if you see tyrosine and tryptophan, they are going to be more polar than phenylalanine. Why? Because the hydroxyl group in tyrosine and the nitrogen in the indole ring of tryptophan, they are going to be slightly more polar compared to the R group of phenylalanine. So tyrosine, tryptophan, and phenylalanine. And phenylalanine, I would say to a lesser extent only, will absorb UV light. So why I am stressing on this particular property is that this gives the characteristic strong absorbance of light by proteins, especially at the wavelength of 280 nanometer that corresponds to the UV region of the electromagnetic spectrum. Okay, so this particular property of the amino acids is being exploited while we need to figure out or find out the concentration of protein in a given sample. So, this graph, as you see, it's a picture from the Lehninger Principles of Biochemistry textbook. So it shows the absorbance that is measured as a function of a wavelength for three amino acids depicted by three different colors. And if you see, the absorbance max is at close to 280 nanometer, and it's mainly due to the tryptophan that is actually contributing to it, and tyrosine to some extent, and phenylalanine is to a lesser extent. So when we kind of exploit this concept, then we have something called the Beer-Lambert's law. This particular law is very, very important because it has been tested in many GATE Excel papers as well as in BT papers. So the Beer-Lambert's law. So there is a particular instrument called the spectrophotometer, which measures the absorption of light to detect and identify compounds as well as to quantify their concentration in a given sample. So if you work in any biochemistry lab, you would have definitely seen this instrument called the spectrophotometer. So this instrument is a very high-end instrument that is designed to measure a light absorption. Okay, so by a given sample that is placed in the cuvette of the spectrophotometer, and upon measuring the absorbance, you will get a clear idea on its concentration as well as even identify what type of compound it is. So the Beer-Lambert's law states that the fraction of the incident light absorbed by a solution at a given wavelength is related to the thickness of the absorbing layer, that is nothing but your path length, and the concentration of the absorbing species. Okay, so it states that the absorbance of the incident light by a given sample is directly proportional to the path length, that is nothing but the thickness of the absorbing layer, as well as the concentration of the absorbing species. So when we put this in a given mathematical equation, you will get this particular formula, that is log I naught by I is equal to epsilon CL. So what is I naught? I naught is nothing but the intensity of the incident light. So when you kind of make a light of a given wavelength incident on a given particular sample, then the intensity of that incident light is denoted by I naught. So what is I? I is nothing but the intensity of transmitted light. Okay, so when the light passes through a given sample, some of them, we know that some of them gets reflected, some of them gets absorbed, some of them gets transmitted. So we are interested in the intensity of the transmitted light, and we denote it by the symbol I. And then there is one particular ratio that is called I by I naught. Here in the formula it is I naught by I, but here it is I by I naught, which is transmittance. So don't get confused and don't kind of mismatch the equation, or mismatch the terms. Okay, and epsilon is nothing but the molar extinction coefficient. So it is nothing but the constant, a kind of constant that you get when you remove the proportionality symbol. So it's called the molar extinction coefficient. It is denoted by liters per mole centimeter. Okay, and then we have C, which is nothing but the concentration of the absorbing species, which is in molarity, so that is uh, mole per liter. And then we have L, that is the path length, which is usually measured in centimeter. So assumptions that we usually take while devising this law is that the incident light is parallel and it is monochromatic. What do you mean by monochromatic? It means that it has only a single wavelength. There is no mixture of wavelengths or any blending. And the solvent and the solute molecules are randomly oriented. So there is not much of interaction or some kind of orientation that is expected. So if you replace this log I naught by I equal to as A, which is nothing but the absorbance, then we write it as A is equal to epsilon CL. Okay, so this law is very, very important because many questions in the past several years have been tested. So thus with the fixed path length A, so if you have a fixed path length, then of course epsilon is going to be constant, you will get A is proportional to C, that is absorbance is proportional to the concentration of the species. So epsilon varies with the nature of absorbing compound, the solvent, the wavelength, the pH. If the light absorbing species is in equilibrium with an ionization state that has different absorbance properties. So if you see the epsilon, which is nothing but your molar coefficient constant or molar extinction coefficient, it is going to vary with the nature of the absorbing compound. It is going to be different for different compounds. It is going to vary with the solvent in which the compound is present. It is going to vary if you're going to change the wavelength of the incident light, and also the pH. If the species that we are actually having it in the as the absorbing species is going to be in an equilibrium with an ionization state that has different absorbance properties. So if the absorbing species has different ionization states, and in a given solvent or in a given condition, only a given ionization state is going to predominate, then even the pH will matter to determine the molar extinction coefficient. So this is again a picture from the Leninger textbook, Principles of Biochemistry, wherein it clearly depicts what this particular law means. You have a lamp, so it is going to emit light, so you need to make sure that the light that passes out and falls onto the absorbing species is going to be just of one wavelength. So it's going to be a monochromator that is going to do this job, so that the light that comes out of it is going to be monochromatic. And this intensity, since this is going to be the incident light, the intensity is denoted by I naught. And here you have the sample cubit, which has a path length, or which has a thickness of L, and that is going to be your path length. And it has the absorbing species. This is the absorbing species whose concentration is C, that is moles per liter, that is molarity. And then there's going to be light, that is going to be some amount of light that is going to be transmitted, whose intensity is measured by I. And you can get the absorbance reading in the detector. And then you can also plot a graph if you like have standards also, and then kind of find the concentration of the unknown species through extrapolation. Okay. Yes. So A versus C you can do, and it's going to be a straight line graph. So this is the standard, and suppose you have an absorbance here corresponding to the unknown. So here you get the concentration of the unknown species. So this is a very simple experiment that we would have done in all our biochemistry labs. Okay, moving on. Okay, now moving on to polar uncharged R groups. So these amino acids are more soluble in water because they're going to be more hydrophilic, and they contain functional groups that even form hydrogen bonds with water because the R group is going to be polar, though they are uncharged. So you have serine, threonine, polarity due to their hydroxyl groups, asparagine, glutamine, and polarity is due to their amide groups, and cysteine, which is actually a weak acid and makes weak hydrogen bonds with oxygen or nitrogen. Why? Mainly because of the sulfur group, and it has the polarity due to the sulfhydryl group. And cysteine, if you see, another interesting property of that is it readily oxidizes to form cysteine. So see here you have two cysteines coming together, I mean furnishing out its hydrogens to form this disulfide linkage, that is forming this structure called cysteine. So two cysteine residues joined by a disulfide bond, and the disulfide linked residues are strongly hydrophobic, and they are going to be non-polar, and it covalently links between parts of a polypeptide chain or different polypeptide chains as well. So this again plays a crucial role in determining the structure of the polypeptide. So if you see negatively charged or acidic R groups, you have two main important amino acids. That's the aspartate or aspartic acid and glutamate or glutamic acid. They have a net negative charge at around say pH 7, that's close to the biological pH, and that is mainly because of the extra carboxyl acid group that is present in both these amino acids. Okay. Moving on, most hydrophilic R groups are those which have either positive or negative charge. Okay, those amino acids that have a positive or negative charge in their R groups, they're going to be more hydrophilic. So the negatively charged we saw, that is the aspartate and glutamate. And now we will move on with the positively charged, that is basic amino acids. You have lysine, arginine, and histidine. So for lysine, it has a second primary amino group at the epsilon position of its aliphatic chain. So you have the last NH3 plus in the R group. And for arginine, it's going to be positively charged guanidinium group. And for histidine, it's going to be its aromatic imidazole group, in which one of the nitrogen actually has a positive charge. So histidine, if you see, it's the only common amino acid having an ionizable side chain with pKa near neutrality. pKa is a measure of how strong or weak an acid is. Okay, so higher the pKa, lower the Ka and lower the acidic strength. So if you see for histidine is close to neutrality, so that's pretty important property to note down. And it can be positively charged, that is in the protonated form, or can be uncharged at pH equal to 7. So why I am stressing on this particular fact is that histidine is one of the important amino acid that plays a crucial role in various enzyme catalyzed reactions by functioning as proton donor or acceptors. So I will be talking about few important enzymes from gate perspective, and I would be definitely mentioning about some role of histidine residue in almost all of them. Okay, so that is why histidine is considered to be a very important amino acid, and these facts have to be in your fingertips. Moving on, there are also some brief notes that I would actually like to mention about certain uncommon amino acids, like the 4-hydroxyproline. It's going to be a derivative of proline. I mean, it's very obvious from the name, and it is found in proteins of plant cell walls, and even it is found in collagen. So when I talk about the structure of collagen, you'll be understanding more about this. Then there is another protein. There's another amino acid that is called 5-hydroxylysine. It is going to be the derivative of lysine, and it is found in collagen as well. And one more is 6N-methyl lysine. It is going to be the constituent of myosin, because these can be tested under certain recall questions of GATE 2024, and it is going to be really helpful, like if you're going to know the answer. So what is myosin? Myosin is a contractile protein of the muscle. So again, this plays a crucial role. And gamma carboxy glutamate, it is present in prothrombin. Anything that is coming ending with some kind of thrombin is going to play a role in the blood clotting protein, blood clotting factors, and in certain calcium binding proteins as well. And there is another important amino acid, that is desmosin. Desmosin is going to be a very complex amino acid because it is a derivative of 4 lysine residues, and it is present in fibrous protein called elastin. There is another two important amino acid. These are called ornithine and citrulline. They don't generally exist as separate amino acid because they are key intermediates in the biosynthesis of arginine, as well as they play a crucial role in urea cycle. I will be discussing about a lot of important notes on ornithine and citrulline when I talk about the biosynthesis of amino acids as well as the urea cycle. So, stay tuned for more videos. Moving on. Yes. Now I'm going to talk about the very important topic, that is the characteristic titration curve of amino acids. We all know that amino acids can act as acids or bases because it has the amino group as well as the acid group. It's amphoteric and ampholytic. So if you see the picture here that I'm just showing here, so this structure is going to be the non-ionic form because there's no charge actually existing here. But if you see here, this is this Zwitter ionic form where there is plus and minus charge, so the electrical neutrality is maintained. But if you see this Zwitter ionic form, it can exist as either a proton donor or a proton acceptor. So here if you see Zwitter ion as an acid, it's going to furnish out the H plus from the NH3 plus group, and it is functioning as an acid because any acid has to dissociate to give hydrogen atoms. I mean hydrogen ions, sorry. But if you see Zwitter ion as a base, it's going to accept hydrogen ions from the solution and form the COOH and gain a net positive charge, and here there is a net negative charge. So that is the reason why amino acids can act as acids or bases. So with this particular concept in mind, I can definitely talk about certain very important concepts pertaining to the titration curve of amino acids. So there's like three important cases that you have to know. So I'm going to talk about the first basic case. So the titration curve of amino acids which are going to have non-ionizable R group, or in other words non-polar R groups, such as glycine. I took glycine as the most important example because that is explained well in all textbooks. So if you see, there are two ionizable groups of glycine, namely the carboxylic acid group and the amino group. So when titrated with a strong base such as NaOH, we all know that sodium hydroxide is a very strong base. So when this glycine solution is going to be titrated with sodium hydroxide, it is going to yield two distinct stages when the titration curve is plotted. So you have like two important stages, 1 and 2. Okay, so I'll talk about what this 1 and 2 are. So if you see at a very low pH, so what do you mean by very low pH? It means that the hydrogen ion concentration is going to be very, very high. So what happens? The glycine is going to be fully protonated. That is, you have NH3 plus CH2 COOH. It's going to be fully protonated, and as a result, the net charge on glycine will be positive. Okay, so when you now add addition of NaOH, or when you now add NaOH, what happens? The carboxyl group of glycine starts to lose its proton. Okay, that's natural. Like when you add a base, the acid will also, it kinds of reacts. So it has to lose its proton to the hydroxyl ion of the base. So what happens at a midpoint of this particular stage, there is going to be equimolar concentration. What do you mean by equimolar? That is the concentration of the species of the different species are going to be the same. So there's kind of equimolar concentration of the proton donor, that is going to be the, the first case where we saw NH3 plus CH2COOH, and the acceptor, and the accept proton acceptor, that is NH3 plus CH2COO minus. So this is going to be the proton acceptor because they have lost their proton. That is only first initially the only the carboxylic acid group is going to lose its proton because that has a greater pKa compared to the NH3 plus. So this is a very logical question if you have in mind, like why only COOH is losing its proton if NH3 plus is already having a charge, a positive charge. That's the case because the COOH group in this glycine amino acid is going to have a lower pKa or higher Ka. I would say it's going to have a greater acidic strength, and hence it loses its proton easily. So there, what happens is a point of inflection is going to be reached, which addresses the midpoint where pH is equal to pKa, which is equal to 2.34. This is the point of inflection because there is not much change in the curve, and this midpoint, if you see, it's going to correspond to a pH which is going to be equal to pKa. This pH is going to be equal to pKa of the COOH group of the glycine. Okay, so this is noted down. So for the first case, now there is going to be another point of inflection as titration proceeds, wherein the removal of the first proton, that is from the COOH group of glycine, is complete, and the removal of the second has just begun. What is the removal of the second proton? It's going to be the NH from the NH3 plus. So this would have just begun. At this pH, glycine is largely present as Zwitter ion because there is going to be COO minus that's already existing, and now this uh, H plus has just started to be removed. So the predominant form is going to be NH3 plus CH2 COO minus. So this is Zwitter ion, right? So this particular Zwitter ion is going to be present in large concentration at this particular pH. That is the reason why we are very much interested on this particular pH, because it gives the measure of pi, that is the isoelectric point of the amino acid glycine. Okay, now let's move on to the second stage of titration, wherein the removal of the proton from the NH3 plus group, and the pH at this midpoint corresponds to pKa of NH3 plus group. So if you see here, again, there's going to be, if you keep on adding a lot of NaOH, what happens? A maximum number of protons will be removed from glycine, and again, the glycine will be existing in this particular form where the net charge is going to be negative. So the hydrogen atom, the hydrogen ion from this NH3 plus group is also going to be removed, and only the COO minus is already present because COOH has lost all its hydrogen ions, and the net charge is going to be negative. Okay, the titration is completed at about pH 12, where the predominant form of glycine is going to be NH2CH2COO minus, which is the negative charge. Okay, so the Pi is equal to the pH at which the electric charge on the amino acid is going to be zero. So what is Pi? Pi is nothing but your isoelectric point, and this is the pH at which the net electric charge on the amino acid is going to be zero. So for amino acids, this particular formula is very important. For amino acids with non-ionizable R groups, or I would rather call non-polar non-polar R groups, the Pi is going to be the average of pK1 and pK2. So uh, these two important data that is being collected from this particular titration curve will be used to find out or measure the Pi of the given amino acid. Okay, so why we are stressing on this particular concept is that in GATE exam you will definitely get direct questions where they ask you find the Pi by giving you the values of pK1 and pK2. Okay, so it's going to be a very simple question. It might even be asked in 2 marks, and you will be doing wonders, like if you can easily get those 2 marks if you're going to know the formula. So this is like pretty simple, for glycine, because you just have two pK values, because there is no ionizable R group in glycine or any non-polar amino acid. But let's see the case with other type of basic and acidic amino acids in the next slide. Okay, so if you see here the case 2 that I've actually mentioned, these amino acids have a negative ionizable R group. So which means that you know they are acidic amino acids, and they are going to be having ionizable R groups that are negative, that bear negative charge. So if you see here the case of the titration curve of glutamate as one such example, if you see, there is going to be three stages in the titration curve rather than two that we saw in glycine. So at very low pH, if you see, the predominant form is going to be, everything is going to be fully protonated. All the carboxylic acid group is going to be protonated, as well as the NH3 plus NH2 is also going to be protonated and has NH3 plus. So the net charge will be +1. So now as you keep on adding, the sodium hydroxide, this particular stage is going to be reached where the net charge is going to be zero. The net electric charge on the amino acid is going to be zero, because this COOH is going to furnish out this H plus to the NaOH. Okay, remember, this COOH is going to do the job first and not the one in the R group. Okay, so make sure that you have that in mind. And now again, there's when you keep on adding more sodium hydroxide, this COOH is also going to furnish out its H plus. So the net charge is going to be -1. And if you keep on adding more and more sodium hydroxide, the proton from the NH3 plus group is also going to be released, and the net charge is going to be -2. So in this case, if you see, if you want to find out, and again, you will measure the pK1, you will measure the pKr. It's a special notation. It's not pK2 and pK3. It's a special notation. And you will also have pK2. Okay, so in this case, when you have to calculate the PI is going to be pK1 plus pKr by 2. Why? Because we have to take that part of the titration curve that involves Zwitter ion. So this part. Okay, so this part. Okay, so it's like these two pKs you have to take, okay, pKr and pK1. So that will have the Zwitter ion in its middle, because that is the stage of the titration where the Zwitter ion is going to be present in its maximum state, and we want that pH, because that pH is going to correspond to the isoelectric point. That is the reason why we have pK1 plus pKr by 2. Okay, but if you see the case of positively charged ionizable R groups amino acids, or I would rather call it basic amino acids, it's going to be a different case. So again, at a very low pH, everything is going to be fully charged, and you will be having a net charge of +2. Let's take histidine as the example. And then if you keep on adding more sodium hydroxide, what happens? This COOH will furnish out its hydrogen ions and starts to gain COO minus, and the net charge is going to be +1. And then again, you keep adding, you will have the net charge is reaching 0. Okay, and then after you go further on, all the H plus is going to be lost, and the net charge is going to be -1. Okay, so what happens is, now we are going to take, if you see the titration curve, it has pK1, pKr, and pK2. Now we have to take that part of the reaction where Zwitter ion is involved. So you will have to take this portion. Okay, so in that case, if you see, the formula to find the PI will be pKr plus pK2 by 2. So remember, here it is pK1 plus pKr by 2, and here it is pKr plus pK2 by 2. So make sure that you are very clear with what type of amino acid it is and what is the formula. Be very sure of the reaction equation, and only take into consideration those values that you're going to use for finding the PI, because if you kind of mismatch it, you're going to lose the marks. Okay, that's the reason. So make sure you're very clear with this particular concept. That is why I am spending one important video on this particular concept itself. Okay, so these are the titration curves, and hope it's pretty clear for you. Yes, this table I decided to put at the last because I have seen few gate papers where they have actually asked the PI of certain amino acids and they gave the PI values as an MCQ question. So if you like kind of know the PI values for all these 20 standard amino acids, I would not say 22. I mean 20 is more than enough. 20 amino acids, then it's going to be a cakewalk question because you can't afford to lose a plus one there. So that is the reason why I've put this table at the last. I mean, you can do it pretty before the exam so that you remember for a long term. That's the reason why I have put it here. So I would suggest all of you to know the proper spellings first, and then the three-letter code, the one-letter code, and then the R group. I mean, here they have mentioned the pKa. I'm not interested in the pKa, but I want the type of R group that is actually present in each and every amino acid along with the API values to be kind of in your fingertips, at least the range, so that it doesn't confuse you. I mean, gate isn't very hard in testing exact values, but it helps you to kind of even score in certain questions so that you have pretty good confidence when you attempt the other tough questions. Okay, so this is the last slide. So I would be posting a few more videos on concepts pertaining to proteins as well. So I would like all of you to watch them and kind of learn really well from these conceptual videos. And before I close down, I would suggest all of you to buy the formula sheet from Miss Priyanka Joshi. I mean, it's only for 99 rupees, and it's a really useful formula sheet, especially for your last minute preparation. I would be emphasizing this on each and every video of mine because it is that very useful. So if you can do it and you use the formula sheet especially for revising before the exam, it's going to really help you because you'll be needing a lot of formula to be remembered in your mind. So I would request all of you, you can get in touch with her through the email ID that is mentioned in the YouTube channel. And you can also get in touch with her through other social media like the Instagram and even LinkedIn as well. So I would request all of you to stay tuned for more interesting content full videos. I would also kindly request all of you to like, share, and subscribe, and kindly subscribe to this YouTube channel as well. Thank you and have a nice day. Happy learning.